Okay, hi everybody. I'm just going to review really quickly a couple of things we covered in the last 3 or 4 slides of class on Monday. It's Monday of week 3. And so that's going to be reverse transcription PCR, quantitative reverse transcription PCR, and then I'll finish up talking about next generation sequencing. So I'm going to do this on my iPad just because I want to draw some stuff. I think that's going to be a little bit better and a little easier to understand. So let's start off with what's called reverse transcription PCR. And this is dependent on the enzyme called reverse transcriptase, which, if you remember, that was discovered by a man called David Baltimore, and it's used to convert RNA into DNA. So this is a little bit foreign, so let's just kind of take it easy and slow to start with. So the normal kind of understanding of the central dogma is that DNA is used as a template to make RNA, and then certain types of that RNA, mRNA, are used to make protein, and then we can think about DNA replication being like this. So the process of reverse transcription takes the information in RNA and uses it to make DNA. So how is this used in RT-PCR? So imagine that you've got some tissue and you want to investigate the level of abundance of, let's just say, actin, or you want to be able to clone an mRNA copy of the actin gene. And the problem with doing that is you can't clone mRNA, so you'd have to make a DNA copy of it. Why would you want to do this? Well, if you kind of think back to what you know about genes, if we have a gene and we have a promoter here, we may have the start of transcription, which we're going to call the +1 site here, and then you would have a series of exons and introns, and if we call this end of transcription, that's the termination site. If we call that down there, we might have exon 1 followed by intron 1 followed by exon 2, and then so on down through the gene. Now, if you're going to try and study a gene, you may not actually want to study all the introns as well as the exons. So what you actually want is a DNA copy of the mRNA, because remember that the mRNA, when it's matured, it's had all of the introns spliced out. So if you can just get a DNA copy of the mature mRNA, that's much more easy to manipulate and work with. So how might you do something like that? So is what we could do is we could take a tissue, for example, and we could homogenize that up and break all the cells open, and then we would put that in a test tube. So let's just have a little test tube here. This is how I draw test tubes. So we've got a test tube here, and in here we would have what is effectively our tissue extract. And so that's all the cells broken up, and so all the cell contents is spilled outside of the cells. And then is what we would add into here would be a poly A primer. And so I'm just specifying that as A with N. Sorry, I need to back up here. That would actually be a poly T primer. You'll see why in a moment. So all that is a long piece of DNA, maybe 30, 40, not really that long, 30 or 40 bases long, and it is just a bunch of T's. So it would look like this, and then the N just represents some number, and that N, let's just say it's an additional 20 T's. So why would we do that? Well, if you think back to what you know about messenger RNA, remember the messenger RNA is going to be in this kind of cell homogenate here. A messenger RNA would have kind of the AUG here, and then the corresponding stop codon would be down here. And as part of mRNA processing, you end up with a run of A's, remember the poly A tail, added to the end of the mRNA. So our primer here of poly T, it can anneal to the poly A tail. So let's just kind of redraw that a little bit so that we move the primer over. So let's put the primer down here. I'm going to take that stop codon out as well for now. So we're just going to move that primer down. So we would end up with the situation where the poly T primer has annealed to the poly A tail. So now, don't forget, this would be the 5' end, and this would be the 3' end. So this is the 5' end of the primer, which means that's the 3' end, which means that, in theory, we could extend the primer towards the 5' end of the mRNA. Now, don't forget that the molecule at the bottom here is RNA, and this is DNA. So the enzyme called reverse transcriptase, it can grab hold of the end of the oligo DT primer, the poly T primer, and it will extend the primer in the 5' to 3' direction so that we make a piece of single-stranded DNA. And then what the enzyme reverse transcriptase can do is it can make a second strand coming in this direction, and I don't really worry about how you do that right now. There are techniques, or there's some complex thing which happens here to do that. And as you do that, is what happens is the RNA template ends up getting degraded, and you have your piece of double-stranded DNA. Let's just get rid of that for now. You end up with a piece of double-stranded DNA. So what you've gone from is you've gone from RNA to DNA. And then you can do the PCR. So what we could then do is we could make a little primer, PCR primer here, and a little PCR primer down here, and we do the heating, the lowering of the temperature, ramping to 72 degrees Celsius. And so if we did that, let's say, 30 times, we would end up with many, many, many copies of the specific cDNA. cDNA is just complementary DNA, it's complementary to the mRNA. So that's how you would do reverse transcription PCR. So that's the first thing. So the second thing that we can think about here would be what's called quantitative RT-PCR. So I'm going to clear my whiteboard here, which is just going to take me a moment. Let's just erase all this, and then we can start over thinking about quantitative RT-PCR. So while I'm erasing this, let's talk about why you might want to do quantitative. Why might you want to quantify this process we just talked about, which is RT-PCR? Why might we want to do that? Let's say we want to compare the level of expression of a gene, and we want to compare it in two tissues to see how abundant the messenger RNA is in those two tissues, so we could make some conclusions about the level of gene expression of a given gene. That's why we might want to use quantitative RT-PCR. So this is going to be the same first step to make the double-stranded DNA from mRNA. So we would have our mRNA here with its poly A tail, so let's just have A to the N. Then we would add in our poly T primer, and we would use that to make a piece of single-stranded DNA, and then the reverse transcriptase would degrade the RNA, and we'd end up with double-stranded DNA. So you would just do that for all of the RNAs you have, because most, if not all, RNAs in eukaryotic cells have these poly A tails. So that's how you do the RT. You do the reverse transcription PCR the same. And so then we would go to the quantification part. So what we do there is we would heat our sample up to separate the two strands of DNA. And we would have our PCR primers, just like we had before. We would draw them in here. But the different thing here is we add what's called a probe. And a probe is a short piece of DNA which can hybridize to our target gene, whatever that might be. And on one end, it has a molecule which is called a fluorophore. So I'm actually going to back up there because I'm going to use a different color. So let's use a different color for this. Let's make that red, and then on the other end we'll make the quench And so the fluorophore here is on one end, and then the quench is on the other. So what is a fluorophore? Fluorophore is a molecule that when you illuminate it with a certain wavelength of light, it will fluoresce. And so if we were to shine some UV light of a certain wavelength on this fluorophore, it would fluoresce, and it would fluoresce whatever wavelength. There are multiples of these you could get, and it would fluoresce. But the fluorescence would be absorbed by the quench. And so the quench is a molecule which absorbs the light that comes off of fluorophore. So when the fluorophore and the quench are close enough together, you won't see any fluorescence if you illuminate the fluorophore. So in this situation, or even if you've just got even if you just have the fluorophore and the quench in solution, which you would have when you're doing the quantitative part of this RT-PCR, any fluorescence from the fluorophore is absorbed by the quench. The only time you'd ever see the fluorescence is if you actually could break the fluorophore off of the quench such that you can actually get them far enough apart. So now if we illuminate this fluorophore, it will fluoresce, but because the quench is all the way over here, because we've separated them, we've broken them apart, we'll actually see the fluorescence. And so this fluorescence here is what we would want to measure. So let me clean up this a little bit, and then we can get back to how this works. So now you kind of got the understanding of the fluorophore and the quench. Let's kind of think about what's going to happen here. So on this bottom strand down here, the polymerase comes along and it extends perfectly happily and gets to the end like that. And so there's a piece of double-stranded DNA. On this top strand, is what we're going to do is we're going to polymerize off the end of that primer. And when the polymerase hits this fluorophore, it is able to cut the fluorophore. It's able to cut the fluorophore off of the quench such that the fluorophore is then going to diffuse away and is freed from the quench. And so the quench will just move away as well. So we can just take the quench away from here, and we can redraw it just over here somewhere. So our quench would end up over here. So now when we shine light on our sample, the fluorophore absorbs it, and then the fluorophore will emit a different wavelength, and it's this that we can measure. And so as the DNA polymerase extends, we get this increase in fluorescence. So when we do this cycle again, if we think about this piece of DNA here, when we heat it up and we anneal it, and our primers attach again, and we've got our fluorophore and our quench, every time you do a round of replication here using the PCR reaction, you release the fluorophore and the fluorescence goes up a little bit. So the more mRNA you start with, the more DNA you have, and the faster you'll get amplification. And so is what you'll see is that if you have, if on this scale it's just an arbitrary unit of fluorescence, and then along the bottom here is the number of cycles of PCR, we just call it number of cycles of PCR. So if you have the situation where you've got kind of relatively large amounts of mRNA to start with, you'll start to get amplification and therefore fluorescence much earlier than if you have very low levels of template RNA to start with. Now you always reach up to the same point. So the number of cycles it takes for you to reach, let's say, this point in the middle, what's going on with the pencil? The number of cycles it takes to reach this point is an indication of how much RNA you use to start with. So that's how you do quantitative RT-PCR, and it's a pretty cool, neat technique you can use to quantify a level of gene expression in multiple tissues. So that's well established and well used right now. Let's do the same thing. I'm going to clean up my slide here, and we've got one last thing that we're going to talk about, and that is going to be next generation sequencing. Your book is going to talk about Sanger sequencing or dideoxy termination sequencing. And to give you an idea, that's what I did when I was an undergraduate student working at the University of Illinois in 1995, 1996. So that's how old that technique is, which is why I don't think there's much use in you learning about it right now. So we're going to talk about what's called next generation sequencing. This is the kind of sequencing that lets you sequence a whole human genome in a few days at this point. In fact, probably about a day. So you'll hear this talked about in research labs, and it's getting cheaper and cheaper and cheaper to do. And it's what will give us the ability to do personalized medicine and start learning about differences in diseases and disease progression in different ethnic groups, and that kind of thing. So this is a little bit complicated, so we'll step through it pretty slowly. There are two parts to it. The first part is called the library, or the sample preparation, or 3 parts, I guess. Then there's a PCR-based amplification of the sample, or the library, and then there's the actual sequencing step. So I'm going to talk you through how this would work, for example, if I wanted to study the types of bacteria that you find in fecal samples of patients who are being treated with 6 different antibiotics, for example. Okay, so if we wanted to learn about how different antibiotics impact gut microflora, this is how I would kind of set up an experiment. So let's keep it simple. Let's just say we've got 2 samples, 2 groups of people who have been treated with different antibiotics. So let's say we've got kind of 2 samples. We'll call them sample A and sample B, and ideally I'd have numerous samples of A and numerous samples of B. Let's just stick with A and B and have 1 of each to start with. So everything I do on A, I'll do on B, and hopefully when we're done, you'll see how you can compare. What you're looking at. So all I need to do is I would take my I don't know. Let's use this emoji. I'll take my poop sample. So I would take my poop sample from A and my poop sample from B, and what I want to know about these poop samples is I want to know what kind of types of bacteria are in the poop samples, and I want to know the relative abundance. Okay, so that's kind of what I want to know about these 2 samples. Why do you use poop samples? You use them because they're reflective of the gut microflora, so that's why they're easy to get. Basically, it's easier than doing, collecting samples directly from the colon, for example, or from the small intestine. You want to go with the colon, but anyway. So here are 2 samples. Let's just focus on sample A, and we're going to take a break here, and then I'm going to start a new recording and post that, just so that these don't get too long. Okay, so I will come back in a moment and we'll start talking about sample A.